So now we will have oral presentations of selected abstracts. We have 7 people that we will be presenting their work. So we'll have 15 minutes talks, and then we'll get 1 question after each talk. And then, if time permits, we'll have more questions at the very end. Our first speaker is Nicholas Cicero, or Cicero.
All right, hello everyone. My name is Nicholas Cicero. I'm a 4th-year PhD student in Laura Lewis's lab, was at BU, is now at MIT. And today I'll be talking about my work studying sleep in humans. So to begin, we're all here because we're interested in discussing things that we do every day of our lives. And I would argue that sleep is one of the things that really is most crucial to all aspects of our lives, beyond the fact that we do it every day of our lives. You probably all heard the fun fact that we sleep for about 1/3 of our lives, and you can appreciate that after a poor night of sleep, our cognition, our mood, our energy is really impacted by this. So much research has really shown this, that both our brain and entire bodies are impacted by sleep pretty vastly. For example, waste clearance in the brain occurs most often during bouts of sleep, with CSF most actively clearing during sleep. Cognitive functioning, such as memory consolidation, occurs during bouts of sleep as well. And mood is also impacted by sleep. Many different mood disorders are characterized by various sleep deficits. Beyond the brain, the entire body, such as various systems within the body, as well as cellular and molecular functions, are also impacted by sleep, really showing how important sleep is. We can also appreciate how important sleep is by seeing how many species across the animal kingdom have sleep-like behaviors. So beyond humans and dogs, various species also show sleep-like behaviors, such as fruit flies, as well as jellyfish. Now, although the exact phenotype of sleep is quite different species to species, as a general behavior, it's quite conserved across evolution. Given that sleep is a pretty strongly conserved behavior, it follows that the neural circuits controlling sleep are also strongly conserved across evolution. And much research in rodents has really found this, that this network of deep brain regions that I'm showing here, which exists across many species, is really vital to regulating vigilance and sleep-wake states. Known as the ascending arousal network, this region has widespread projections throughout the brain and releases neuromodulators pretty diffusely across large-scale brain networks. And these are just a handful of those neuromodulatory systems. One of the downsides of these rodent papers is that they usually target each region one by one, either stimulating or recording from these regions and observing how that impacted sleep-wake states. But we know that this is really a network of regions that has some sort of coordinated activity, and to really understand how this network is functioning as a network, we really need to observe this network simultaneously, which is one of the advantages of the human neuroimaging approaches that we have today. We also want to see how these subcortical regions might regulate arousal and the dynamics that occur across arousal. If we look at a EEG spectrogram during a night of sleep, although we can score these stages of sleep pretty discretely, we can also see that there's a lot of dynamic fluctuations occurring that don't always fit into one single category. Additionally, the ability to behave when you're in a drowsy state is not always a kind of binary yes or no. It's very dynamic. So to understand this question of how these deep brain regions might regulate arousal, we also need a multimodal approach where we collect not just MRI, but also behavior as well as EEG. So that brings me to my PhD work, in which we're using high-resolution fMRI simultaneous with EEG to investigate this network. To briefly talk about some of the limitations in conventional neuroimaging that have limited the study of this network so far, firstly, the regions that are in this network are really small. So if you haven't heard of these regions before, I can just talk about one brief example. The locus coeruleus is one of the primary noradrenergic regions and sits very close to the fourth ventricle. In post-mortem studies, it has been shown that this region in its smallest direction is about 1 to 2 millimeters big. Conventional fMRI has about 2.5 to 3 millimeter resolution, which vastly is too big to really resolve activity in these small regions. So if we really want to understand activity in these small regions, we need to push the resolution of our sequences to a much more accurate size to these small regions. So with things like 7 Tesla MRI, we can actually begin to push the resolution to a size that more closely matches these regions while still maintaining a high SNR. Additionally, EEG is really the gold standard to observe and quantify different sleep behaviors, but doing EEG inside of the 7 Tesla scanner is quite new. There's a lot of safety concerns that it brings with because of the different magnetic or metal components that are inside of the 7 Tesla scanner. And doing simultaneous EEG in the scanner also results in artifacts in both the EEG data and MRI images. Luckily, we have a collaborator at Martino Center who has developed this new EEG net that has no copper wires. It's actually conductive ink that collects the signal. So we actually have a paper coming out pretty soon, hopefully, that shows that this is both safe to use at 7T and also reduces image artifacts in MRI scans, and also has a lot of clinical applications. So we're going to use this cap in our study to also get past this limitation. So that brings me to my experiment we're doing, which we bring subjects to the scanner around the typical bedtime, and we do simultaneous EEG, fMRI at 7 Tesla. When they're inside the scanner, we instruct them that they are allowed to fall asleep, and that their only task is to do this self-paced button press task where they press a button every second. This is just a way for us to measure if they are responsive or not. And they're inside the scanner for up to 4 hours. While they're doing this scan, we are collecting this high-resolution functional sequence that we developed to have a spatial resolution that much more closely matches these deep brain regions. So we have a 1.2 millimeter voxel size, and we're still sampling below a second. The middle grid there shows the field of view outlined in blue, and the bottom is just a GIF of that high-resolution data. So we have data from 20 subjects. On average, each person has about 2.5 hours of data per session, and we did have 3 subjects come back for a second night of scanning. Our EEG data we did sleep scoring on with clinical criteria, and we observed that over half of our data was from early stages of non-REM sleep, and then the rest was during primarily wakefulness. We didn't really get much deep sleep for reasons I could talk about afterwards. To briefly show some of this multimodal data, this is just a snippet over a few minutes. So we have our EEG spectrogram that we can do sleep scoring from, and then in the middle we have our fMRI data across these various brainstem regions. We have our behavior, which I'm plotting here, each button press as a circle. And then we also have several measures of physiology. So this is really a wealth of data to work with to look at these various dynamics. Firstly, we were interested in looking at how these subcortical regions might be activating or changing activity when people are going between different stages of sleep. So based on a previous study in our lab that had a very similar paradigm, we defined a behavioral arousal as the first button press after at least 20 seconds of unresponsiveness. So you might be wondering, were they just forgetting to press the button and then remembered? So with our EEG data, we can really confirm that they truly were asleep. So with this, here I'm showing the behavioral arousal as the white dashed line. And in the EEG, you can see that when this arousal occurs, there is an increase in power in this 10 to 12 hertz frequency. So this occipital alpha power is associated with wakefulness. So this really shows that this person wasn't just forgetting to press the button, but they truly were asleep and then woke up. If we average this alpha power across all arousals, we can see that there's a really strong peak in alpha at this behavioral moment in time, suggesting that this really is a sleep-wake transition. So from that previous study in the lab, they found that the thalamus had an early rise in activity before the arousal, which I'm denoting here by the dashed line, whereas the cortex had a strong deactivation after the arousal. We're seeing a very similar thing to what was shown in our previous paper, but I guess more interestingly, and the point of this study was to look at the deep brain regions. So most of these regions have been shown in rodents to promote wakefulness. So we would expect that they would activate at this moment in time. In this single region, locus coeruleus, we do see that, where the LC has this early rise in activity that peaks a little bit after the arousal moment. And then across all of that those ascending arousal network regions, we see a very similar pattern where most of these regions are activating and beginning to activate before the arousal occurs. You might be wondering what's going on here. So the preoptic area is actually one of the few regions known to be sleep promoting. So it's actually more active during sleep than during wakefulness. So we nicely see here a correspondence with that result where the preoptic area actually deactivates when people wake up. Next, we wanted to look at the other side of the transition, so when people were falling asleep. In general, sleep onset is pretty hard to define. It's not always an obvious moment in time. So as a proxy measure, we defined arousal dip as the last button press before subjects became unresponsive. Again, with EEG, we can confirm that this really was a change in arousal state. So we could see in this example trial that the alpha power, which signifies wake, is decreasing after the arousal dip. And then across all arousal dips, alpha has a really strong decrease. So this truly is a wake to sleep transition. So whereas the thalamus and cortex, whereas the thalamus activated and cortex deactivated during arousal, we see a nice kind of opposite result here, where the thalamus actually deactivates as subjects fall asleep, whereas the cortex has a later activation. And if we overlay those two results, we can really nicely see the contrast between those different types of arousal transitions. Now, across the ascending arousal network, we see a very similar thing, where these regions that were known to be wake-promoting now have a dip in activity as subjects fall asleep. And again, we can overlay the response and really nicely see kind of the contrast in response between arousal dip and the arousal. So none of that was really new. It was a lot of things that had been shown in rodents, but just hasn't really been confirmed in humans yet. So that was a nice replication of what we expected to see. Next, we wanted to see if there was different kinds of arousal events that were occurring. So for example, sometimes people would have an arousal event and they would stay awake for quite some time. Other times they would wake up and shortly fall back asleep. So we were curious if these different kinds of arousals, that kind of reflecting the maintenance of awake state, actually had meaningful brainstem activity to correspond to that difference. So as I mentioned, sometimes we observe these transient arousals, sometimes we observe these sustained arousals, and when we group the arousals into these different subtypes, first the EEG alpha power corresponded with this, where the sustained arousals had sustained alpha, showing a true kind of difference in arousal state. And then when we looked at the fMRI data, we were intrigued to see that only this subset of regions actually reflected this difference in behavior. This was really surprising to us because this network has really been thought to be a pretty kind of cohesive, homogeneous network. But this suggests that there might be some heterogeneity with which regions are actually reflecting different kinds of arousal events. And lastly, we also separated arousals by how long the sleep period was before people woke up. So for example, an arousal from deep was at least a minute of unresponsiveness before the awakening, whereas the arousal from light was a much more drowsy kind of in and out of sleep period. And we hypothesized that to awake from a longer sleep period, you might need a stronger arousal response from this subcortical network. So we found that this other subset of regions really now reflected this sort of behaviorally meaningful difference. And most of these subcortical regions had a much stronger amplitude activation when the arousal followed a long sleep period. So this result, paired with the last result, really shows that there's heterogeneity in this network that has some really meaningful behavioral relevance. So in conclusion, we were able to use high-resolution fMRI to measure dynamics in these deep brain regions in humans. We found that regions within this ascending arousal network activated before arousal transitions occurred and deactivated following dips in arousal. And then we also found that different kinds of arousal events showed that subsets of regions within this network reflected these differences. So really suggesting there's some heterogeneity in this network with some meaningful behavioral differences. And in general, this is certainly not the first study to do this, but this is one of the studies that's really is beginning to push the resolution of these fMRI sequences. These deep brain regions are important for widespread behavior, so I think this can be improved upon in trying to get more high-resolution imaging of these really important regions. And again, this is not the first study to do it, but kind of one of a handful that are beginning to push the limits of this multimodal imaging at these higher magnetic field strengths. So with that, I want to thank everyone in the Lewis Lab, our amazing collaborators at Martino Center, the BU Grad Program for Neuroscience, as well as our funding sources. Thank you.
Thank you so much. And we have time for one question from the audience. That was good. Great, thank you. Our next speaker is Sein Jeung.
Okay. Hi, everyone. And first of all, thank you so much, the organizers and the program committee, for giving me the opportunity to present this project. And the title of the project is somewhat cryptic, but I'm glad that I can explain this in more detail with visual aid. So the basic goal of this project, which is a collaboration between two labs, one in Berlin Brain-Body Imaging Lab, led by Klaus Graumann, and then Christian Döller Lab, based in Leipzig, Germany. The aim of this project is to see if we can actually capture activities in electrophysiology that originate from deeper sources. And to this end, we try to test models that have been established in other neuroimaging methods and is informed by animal research. Hexagons are actually quite special shapes because in nature, it is the optimal solution for the close packing problem, where you have to squeeze in as many circles as possible within a limited area. And this comes up occasionally in nature as well, as, for example, as seen in beehives. And this kind of geometry is also observed in a certain cell group in the entorhinal cortex. So this morning in Nantia's talk, we saw that medial temporal lobe of rodents and other mammals house these cells that encode space in which the animal is navigating. And the entorhinal cortex houses grid cells that has a characteristic pattern that tessellates the space in triangular grid shapes. So first observed invasively in single cell recording in rodents, you see that the firing fields of these grid cells have even coverage of the space. And if you connect the center of the firing field of one cell with the adjacent centers of other firing fields, then this forms a hexagonal shape. The thing is, on the macroscopic or mesoscopic level, this signal is difficult to capture when an animal is staying at a fixed location, because individual grid cells have phases that are slightly shifted from one another. So if you look at the figure on the top left side, you see when they are superimposed on top of each other, there will be no possible signal that we can read out when an animal is fixed at a location. So the good thing is that grid cells are organized according to other types of principles. So the first thing is that they are organized into different modules that represent different spatial scales. And by analogy, it is similar to how we represent time in different scales, such as seconds, minutes, hours, days, and weeks. So different grid cell modules cover different types of spatial scales. And then, as I mentioned, the grid phase differs between different neurons. But the good thing is that grid orientation, which is the orientation anchoring to the allocentric environment, tends to be relatively consistent or coherent across different cells. And this inspired this study in 2010, where it was attempted to identify whether humans also have grid cell-like system, and this was done using fMRI. So the idea is that when an animal or a human translates aligned with the main axis of the grid cell-like template, then this should maximize or minimize the neural activity, in this case the ball signal. And then when the animal is walking or translating apart, 30 degrees apart from that main axis, then the activity will dip. And then if it's 60 degrees apart, then the activity will peak again. So the exact alignment of the peak and troughs of that modulation is still debated. But the gist is that you will see this kind of six-fold modulation by the orientation of the animal's translation reflected in the Ball signal. And surprisingly, this worked in MRI study where participants were lying inside a scanner and did the first-person perspective navigation using joystick. So the Ball signal in the aligned runs were higher versus misaligned runs. And this has been replicated in multiple other fMRI studies as well as IEEG and even MEG studies. And the fascinating thing is that this effect generalizes to non-spatial spaces such as mental simulation, imagination, and concept spaces, where you map features of the stimuli in two-dimensional abstract concept space. It also seems to recruit the same entorhinal network. And this might be the key to understanding the unique aspects about human cognition that is not seen in other animals. And it would make sense for evolution to make use of the system that already exists in the brain that's already representing physical space to represent abstract or concept space. So what's missing here is, of course, that humans, as we heard from Nantia's talk earlier, the rodents or animals that are used in single-cell recording are moving around this space using physical motion, whereas humans are confined in the stationary setup in these setups. So this could actually give us the means to approach the medial temporal source activity, maybe non-invasively and even in mobile applications. So the data set that I'm presenting today is recorded in stationary setup, but this is what I see as the significance of testing this computational model or analysis method in scalp EEG. So yeah, basically we want to generalize the findings or what we know about rodent medial temporal system and then ask what happens in the grid cell system in humans when we are walking outside in the real world. And this six-fold symmetry analysis could be the link that bridges this gap. So what we tried in this stationary experiment is to equip participants with high-density EEG. Originally, this was planned to be followed up with a mobile EEG component. So that's why the virtual environment is rectangular, which is kind of a double-sized replica of our lab space. And participant is equipped with mobile EEG system. And in this proof of concept study, we let the participants navigate using keyboard with the first-person perspective 3D environment. And for capturing this six-fold modulation effect, we extracted EEG signals from those segments where participants are translating with the heading maintained for more than 500 milliseconds. And then we source localized to capture the deep source activity from scalp EEG signal. So to this end, we divided the EEG signal into 5 different frequency bands, ranging from theta, alpha, beta, and low and high gamma. And then we fit the generalized linear model with regressors that represent the sine and cosine of the 6 times the angle of the instantaneous heading of the participant. And the good thing about this GLM is that when you fit the model to the data and then extract the beta parameters and then take the inverse tangent of this beta, then it estimates the phase of the sinusoid modulation to the data. So because the modulation of the grid cell activity is anchored at a certain orientation in the environment, and we don't really know how it's anchored with respect to the environment, we kind of need to figure this out from the data. So that is the purpose of this GLM, and we used cross-validation method where we divided the entire data into 5 different sets, and in 4/5 or in 80% of the data, we fit this GLM to estimate the phase first. And then in the remaining in the remaining 20% of the data, we estimate the magnitude of this modulation. So we extract the beta parameters of that second GLM, where the heading value estimated from the first GLM is fitted to the data. And then as a conservative statistics, we created a surrogate distribution of beta values using the same procedure, but with shuffled headings. So the mapping between the headings and the EEG power values were shuffled. That way we can tell like how much of the activity is specific to the 6-fold modulation. And we also included 4, 5, and 7, 8-fold modulation as control symmetries that are biologically implausible. And this is the impression we get with the parameter space that we searched for. So the 5 frequency bands and 4 different region of interest masks, source localized. And as you can see, the z-score of these beta values against surrogate distribution is highest in the 6-fold condition in high gamma band. And we do see some activity in the low gamma band as well. And when we run the t-test, these are the effects that survive the multiple comparison corrections. So left parahippocampus, parahippocampal cortex, high gamma, and then left hippocampus, low gamma, and right hippocampus, high gamma, and parahippocampal cortex, high and low gamma. I was asking myself how meaningful this is to look at deep source activity using scalp EEG, like how well the source localization work, because yeah, the spatial extent of the signal is pretty broad. So with that results of t-tests, we extracted the heading of the conditions where the effect was significant, and then divided the trials into aligned and misaligned runs. And if we contrast that, we can actually see the topography of the effect, where it's pronounced in 6-fold high gamma condition. But if you look at the channel level activity, the histogram of the t-values, the 2 distributions, 4-fold and 6-fold symmetry, are not really separated from each other. And what you can also see is the theta 6-fold symmetry, which did not show up as significant in the t-test, also shows some kind of effect. But this is, again, like not really specific to 6-fold. I still don't really know why this is the case, but at least for the high gamma band 6-fold symmetry, channel level doesn't seem to be the ideal approach. And when we go into source level, you can also see that the spatial effect is super broad, but the voxel-wise distribution is better separated between 4-fold and 6-fold symmetry. And we still do see something in the theta band as well. So as I asked earlier, my biggest question is like whether this gamma oscillation that we see is actually coming from entorhinal cortex proper, or is it propagation to some other brain areas? But at least it seems to align with the finding from the MEG study from 2019. So yeah, we then did the analysis on the heading estimates, the potential alignment of the grid cell-like template to the allocentric environment. And this really aligns well with the previous findings from IEEG or MRI literature, where the phase estimates are coherent across different regions of interest, but they vary across participants. And the correlation with performance has a negative going trend, but the regression wasn't significant. But then if we divide participants into participants for whom the Z grid score was non-significant versus significant, the participants for whom the modulation was significant tends to perform worse. To sum up, the significance of this work is that we identified the analysis variant that works for scalp recorded EEG data. So now that we know which frequency band to look at and what kind of methods to use to capture this effect in scalp EEG, we can probably bring this to a mobile setup. And we do need to polish the methods to be able to use in situations with even lower signal-to-noise ratio. But then another aspect of mobile recording is that we don't really know if the signal itself will be stronger. So it is possible, as seen in animal research, when people can move in physical space, the signal itself can be stronger. And then we see mixed results about correlation with performance. And this seems to have something to do with the fit between the environment or task demand and the participants' navigation strategy. So there's still a lot of unknown things and question marks, but I hope that as the field advances, it will be addressed in the future, and we'll be able to capture the medial temporal activity in humans as well. Yeah. The acknowledgement slides.
Any questions? Okay, thank you very much. I will move to our next speaker, which is Lara Papin.
Hi. I would also like to thank you for the opportunity to speak today. So my name is Lara. I'm a PhD student from Germany, and my lab is mainly interested in bringing EEG into the real life. And this is a typical real-life example. You probably all know it. It's a walk and talk paradigm. And what we call this is cognitive motor dual tasking. And this is also what I am mainly interested in. So at what happens when we concurrently perform a cognitive task and a motor task. And there has, of course, been some prior research on this, and various things have been found. And one is that the performance in either one or both tasks may be modulated when performed concurrently. And this modulation might look differently. So in this walk and talk paradigm, for example, some people may just stop walking when they start to talk. This is a typical example for what we call cognitive motor interference. This is believed to be the case because attentional resources are not unlimited, but they're rather limited. So we have a certain amount of attentional resources, and as soon as the upper threshold is hit, they may be allocated to a certain task. In this case, with the walk and talk, when people stop walking, it's allocated to the cognitive task, to the talking. And we heard about this yesterday before about the P300, that the P300 could, for example, also be used in BCIs because it marks attention someone pays to a certain thing, and it also marks the allocation of attentional resources. And in order to measure an event-related potential, we of course need EEG, and in our case, it's mobile EEG we use because we, as I said already, want to move EEG into the real life. And this is what a typical EEG setup we have could look like. So participants are wearing a mobile EEG cap, rather minimal, so not too many electrodes. Then we have in the back, this white thing is the amplifier. It streams the signal onto a smartphone where the signal is acquired. And since we work with a lot of auditory paradigms, people also wear headphones or earphones a lot. And okay, now we have the EEG setup. Now we need to provoke our P300 in order to measure it. And a very good way of doing that is an oddball paradigm. This is an example of an oddball paradigm. We have two different tones. One occurs frequently, which is here the gray tone, and one occurs infrequently. And we then ask the participants to react to the infrequently occurring tone by, for example, either counting it or pressing a button every time it occurs. So the gray tones are ignored by the participants, and the blue tones are paid attention to. And what then happens in our EEG is something like this. So we have tone onset where the blue note is displayed, and then we have a nice ERP response. First, we have a negative deflection after about 100 milliseconds, which is the N100. This marks, okay, I heard the tone. So it marks that participant actually received the tone. And then what we are mainly interested in here is the P300, which is then this large positive deflection after about 300 milliseconds. And that marks, okay, I did not only hear the tone, but I also paid attention to it. So this is our attention marker. And now when attention is allocated, because the second task comes into play, for example, walking, two things may happen. First of all, it may be modulated on the y-axis in amplitude, or it may be modulated on the x-axis in latency. And what typically happens when we see cognitive motor interference is this. So it drops in amplitude, and it shifts or it shifts in latency. So we see it later in the EEG. Okay, this is a very nice picture, but this is of course not real data, so I'm going to show you how this now looks like in real data, in cycling, first of all. So here we have the single tasking condition. All of the attentional resources that are needed for the oddball paradigm are available and are used for the paradigm, which is why we have P300. Here it's negative because the y-axis is flipped, but we have a relatively large P300 marked in blue. And what's also very important is that this P300 only occurs after deviant tones are played, not after standard tones are played, but this is also what you would expect. Now, when people dual task, so when they cycle while they complete the oddball paradigm, we see a reduction in amplitude. And this is not only the case for cycling outdoors, but it's actually also the case for walking outdoors. So here in red, we have walking and oddball paradigm at the same time, our dual task. And in blue, we have the single task, which is standing and performing the cognitive task, so the oddball. All right, so this so far is the prior research that has been done. But I initially started with this picture with a nice walk and talk paradigm, and you would all agree that oddball isn't quite a good representation of talking. However, one crucial component of conversing is listening. And the other thing about this paradigm is that walking and also cycling introduce a lot of artifact into the EEG. And we don't really know where and why here the interference happens, because in walking we have a lot of different things that are going on. We not only have a balancing part, for example, but we also have visual flow because we're moving through the room. And this is why we had the idea, one of our colleagues was slacklining a lot, and slacklining is a complex balancing task. You probably all agree on that. We had the idea, okay, let's look at one crucial component and let's see if this component is actually responsible for the cognitive motor interference effects that have been seen in walking before. So balancing is one crucial component, as I said already. So we asked ourselves the question, do complex balancing, as in slacklining, and the ability to attentively listen to sounds influence each other? And in order to assess this, we put our participants on a slack line. So the green line you see here is a slack line. Our participant is wearing the EEG device on their head. They're also wearing earphones. You probably can't see that right now. But they were actually just supposed to stand on the slack line, so they didn't walk. They were allowed to move their feet one forward and one backward, so that was fine. But anything else wasn't allowed because we were very interested in this balancing purely. Okay, so in total we had 3 different conditions, 2 single tasking condition, one was a single motor condition, which you can see on the very right. One was a single cognitive condition, so people were standing with oddball presentation, and one was a dual tasking condition where people were balancing on a slack line and listened to the oddball at the same time. And because people were not able to stand on the slack line for a very long time, because it's very, enduring and a lot to put on our participants, we asked them to balance on the slack line for 2 minutes in a row and then repeated this whole process 6 times in total. We also recorded a baseline, and in addition to the EEG, we also had some motion sensors we put on our participants to see how they were moving, but I'm not concentrating on motion here, I'm rather concentrating on the EEG signal. Okay, so everything's set up. What happened to our P300 now? All right. In total, we had 18 participants, and what you will see in a second is a grand average across all of these 18 participants. This is the single tasking condition, single cognitive tasking condition, where people were standing. So you can already see, in response to deviant tones, we have a beautiful P300. In response to standard tones, we don't see a P300. The topography you see on the left is also very typical. So it's rather at parietal channels. And this is what it looked like for slacklining. So not real difference on first glance, right? Okay, why could that be? This was the next question we asked ourselves. First, it could, of course, be because we don't have the visual flow we have in working, for example, but we just have a single balancing paradigm. But then we further looked at individual data, and this is what we saw. So here on the left, you can see the both amplitudes in response to deviant tones, and each dot represents one single participant. And you can already see that it's spreading rather wide here. And what you may also see is that the gray lines, which indicate the change of the P300 amplitude between slacklining and standing, go down for some people and go up for other people. So some people actually had a higher P300 amplitude in the slacklining, which is kind of unexpected, because we actually expected that it would be higher in standing compared to slacklining. The same was the case for the latency. So for some people, the P300 occurred earlier in the slacklining condition. For some people, it occurred earlier in the standing condition. However, on group level, the latency as well as the amplitudes were not different between the two movement types here, so between slacklining and standing. So we thought to ourselves, okay, one of the major reasons could be skill level. So people who have been practicing a lot of the slackline are just more skilled, and the movement is just way more automatic for them compared to people who don't have a very high skill level, where it probably requires more cognitive resources. I think we heard about this yesterday with walking as well. For healthy people, walking is usually a very automatic movement, while for people who have some disabilities, it's rather a cognitive process that's behind the gait. So what we did was we looked at whether the change in the amplitude, so slacklining amplitude minus standing amplitude, correlated with skill. And you can see here on the, maybe I have, yeah, I do. So you can see here on the upper end, these were the people who had a higher amplitude in the slacklining condition, and these here were the people who had a higher amplitude in the standing condition. So this is what we actually expected. And for amplitude, this didn't correlate with skill. I mean, you can probably see a clear trend, but it didn't really correlate significantly. But for the latency, it did. So here, it's a little irritating because it's the other way around. And this isn't there. So here on the top, we have people who had the P300 earlier in standing, and here we have people who had the P300 earlier in slacklining. And this indicates that people with a very high skill level showed facilitation effects. So they had more resources on the cognitive task during slacklining than they did during standing, while people with a low skill level had rather interference effects. So they had the opposite effect that people with a high skill level showed. Okay, so to conclude, well, we were actually able to obtain meaningful mobile EEG results in a naturalistic setting. And here you can already see one guy that is juggling. He had a very high skill level, so maybe the video is working. Yeah. So here you can see someone with a very high skill level. He actually triple tasked easily. And still he was able to perform the task quite well. So this tells us that it's very important to take skill into consideration when we look at motor tasks that have a wide variety of skill levels. And this brings me to my final figure. Here we have a very nice figure of different effects that can happen when we introduce two different tasks to our participants. So we do not only have this cognitive motor interference, which is here, but we also may see some cognitive motor facilitation effects, or just motor interference, or just motor facilitation, for example. Okay. With that, I would like to thank you for listening, and my co-authors and my whole team back in Oldenburg for helping me with this study.
Thank you so much, Lara. Do we have any questions from the audience?
Thank you for the wonderful talk. I was just wondering, was the oddball presentation something you could easily change the difficulty of? And have you tried looking into that to kind of elicit a cognitive motor interference effect?
Can you come again with the last bit?
So basically, did you try making the oddball presentation harder to make the interference happen?
So we didn't do that, but there are studies on that. And what you typically would do is you would, for example, increase or decrease the difference between the tones. So here we had one 800 hertz and one 1000 hertz tone, but if you move them closer together, it gets harder for the participants.
Right.
And it actually also modulates the P300 if you do that. Yeah.
Okay, thank you.
Thanks for your question. All right.
Thank you again. Our next speaker is John McLinden.
Okay, hello everyone. I'm John McLinden. I'm a PhD candidate working under Dr. Yalda Shahriari in the Neural PC lab at the University of Rhode Island, and I wanted to share a little bit about the work we've been doing on investigating causal interactions between EEG and FNIRS during auditory processing. All right, so multimodal neuroimaging, as you guys are probably aware by now, refers to the concurrent use of two or more neuroimaging modalities, and one of the most common non-invasive neuroimaging multimodal neuroimaging configurations is the concurrent use of EEG and FNIRS. So EEG measures the fast electrocortical responses in neural activity, while FNIRS measures, by comparison, relatively slower hemodynamic responses. Both of these modalities can provide complementary information about underlying neural function and also be potentially quite stylish. So we're interested in multimodal neuroimaging during the auditory steady-state response. So to elicit this response, we use basically steady-state white noise click trains that are presented to participants. When participants perceive these stimuli, they produce an auditory steady-state response at the frequency of stimulation. So in our case, we're interested in the 40 hertz steady state response, auditory steady state response, which is produced primarily by the left and right auditory cortices, among other subcortical structures that we're not able to image with these non-invasive modalities. When we presented these auditory stimuli in blocks, we were able to observe a hemodynamic response in the left and right auditory cortices associated with presentation of these stimuli. So while we were able to find some complementary information about the generation of the ASSR, we were interested in potential causal interactions between EEG and FNIRS. To do this, we investigated the full frequency directed transfer function, or FFDTF. So the FFDTF is a causal metric from the family of methods relying on multivariate autoregressive modeling as an extension of Granger causality. So after proper data conditioning for both modalities, we can concatenate these two signals from EEG and FNIRS and fit a multivariate autoregressive model to both signal types. And once we've fit that model, we're able to extract the transfer function matrix of that of the weight matrix here denoted A sub k as the inverse of the Fourier transform of that estimated weight matrix. From there, we're able to extract the full frequency directed transfer function, which measures causal interactions between two of the variables. So for our experimental design, we recruited 17 healthy participants to participate in our experimental task, which consisted of 3 runs in which 24 blocks of auditory click trains were presented in 15-second blocks, followed by 15-second resting state blocks. And we recorded EEG and FNIRS simultaneously from 3 separate regions of interest, those being the frontal, frontal-central, left auditory, and right auditory regions of interest. To cover how we preprocessed our signals, we had to kind of condition EEG and FNIRS in a way such that fitting both of them into a multivariate autoregressive model would make sense. So what we did was, for EEG, we narrowband filtered each of the EEG signals into 3 separate frequency bands of interest, those being the alpha and beta bands, along with a narrow band around the frequency of stimulation, which we refer to as the ASSR band. And we extracted the envelope of the Hilbert transform of these narrowband filtered signals. For FNIRS, we followed a relatively standard preprocessing pipeline including conversion from optical density to oxygenated hemoglobin concentration change using the modified Beer-Lambert law. From there, we were able to concatenate these 2 signals and apply some common data conditioning techniques, including bandpass filtering, resampling to 4 hertz, visually identifying and removing bad channels and trials from the data sets, averaging the remaining channels over regions of interest, within trial z-scoring, detrending the signals, and subtracting the ensemble average before fitting these to a multivariate autoregressive model. We fit these MVAR models to the task and rest blocks individually, as we're interested in comparing the 2. We selected a model order of 6 corresponding with 1.5 seconds of data based on minimizing the Bayesian information criterion. And we extracted FFDTF as described previously. We were interested in determining if there was a difference in the auditory task and cross-modal FFDTF means. So we were interested in comparing the extracted FFDTF across our task conditions. To do so, we applied a permutation clustering-based approach along adjacent frequency bins in this FFDTF metric, and comparing it with a surrogate distribution generated by shuffling condition labels across participants. We do want to note that we were only interested in investigating the cross-modal FFDTF values, so we didn't include the generated within modality FFDTF values in this analysis. So after doing all this, we observed 3 significant clusters. The first is higher front-to-central ASSR EEG, so EEG in the ASSR band in the direction of left auditory FNIRS, FFDTF values during the task relative to rest. We also observed lower FFDTF during the task relative to rest in the direction originating from left auditory FNIRS to right auditory alpha band activity, and from right auditory FNIRS to right auditory ASSR activity, EEG activity. So the higher front-to-central ASSR EEG to FNIRS left auditory FFDTF during the task relative to rest to a degree makes sense. So when we looked at our unimodal results, we observed the ASSR maximally in the front-to-central region in EEG, while we observed hemodynamic responses in the left and right auditory cortices. And as expected, the ASSR preceded the hemodynamic responses temporally. So in that way, this cluster makes sense. The other 2 clusters are a little more difficult to interpret. So as I kind of alluded to just now, any electrophysiological response and coinciding hemodynamic response would basically follow in the direction of the electrophysiological response would precede any associated hemodynamic response. But we observed a couple of clusters in the direction originating from FNIRS to EEG variables. Now, these could be related to ongoing hemodynamic processes, modulating activity in EEG that changes with the task, but this interpretation is fairly preliminary. In the case specifically of the left auditory to right auditory FNIRS to alpha EEG being lower during the task relative to rest, this could be related to attention, but we would likely require broader sensor coverage to be able to investigate that claim further. And in the case of the right auditory FNIRS to right auditory EEG ASSR FFDTF being lower, we would potentially attribute that to disruptions of ongoing relationships between EEG and FNIRS due to the sudden presence of auditory steady-state responses in EEG, which we would need to potentially investigate a little further using a higher model order, looking further back into the data. And that brings us to 2 of the challenges that we hope to address in the future and to improve this kind of analysis framework. The first challenge that we want to address is that challenge of selecting an appropriate model order. So as I mentioned previously, we were using a model order of 6, which corresponds with a second and a half of data, which likely isn't long enough to fully capture the interactions between EEG and FNIRS. At that time scale, an order of 6, a model order corresponding with about 5 seconds might make a little more sense and be able to more fully capture these relationships. But this comes with a trade-off in that it increases model complexity and overfitting. So we're looking for the best way to kind of address that trade-off. The other challenge that we plan on addressing is the influence of systemic physiology in FNIRS. So FNIRS signals contain, especially long separation FNIRS signals contain extracerebral hemodynamic components and other sources of physiological interference, which we didn't account for in this analysis. We did record a set of short separation channels along with these long separation FNIRS channels as independent measurements of these systemic sources of physiological interference in FNIRS, but we're still looking for the best way to include these in our analysis framework, either through including them as additional inputs to the MVAR model or potentially through a regression and subtraction-based method. We're still investigating the best way to kind of approach that problem. So at this point, I would like to acknowledge our funding sources and thank you for your time. Really appreciate it.
Thank you, John. Do we have any questions? Anyone from online? Yes.
Could you elaborate a little bit on extracerebral?
Oh, yes, definitely. So in FNIRS, maybe I can, if I can get back to my slides. If not, that's okay. Yeah, so in long separation FNIRS signals, basically, when you record long separation FNIRS channels, you're passing through, there we go. When we emit near-infrared light into the surface of the cortex, it also passes through several layers of non-cerebral tissue, including the skull, the scalp, CSF, and there are signal components related to those, including extracerebral hemodynamic cilic, scalp vasculature, that are captured and kind of generally represent global sources of variance in FNIRS signals. So it's become standard practice at this point to account for those using short separation channels that measure that variant, those signal components independently. So a really good way to approach that in a more standard FNIRS processing pipeline is probably like the work on temporally embedded canonical correlation analysis and generalized linear model framework developed by Dr. von Lumen, which, good work on that. That was awesome.
Thank you. Thank you so much again. Our next speaker is Kevin Ramirez-Chavez. Welcome, Kevin.
Okay, hello everyone. My name is Kevin Ramirez-Chavez. I am a PhD candidate at Drexel University and a member of the Neuroergonomics and Neuroengineering Lab led by Dr. Hassan Ayaz. And so I'm really excited and honored to present my work titled The Psychophysiological Responses in Virtual and Real Environments, a Multimodal Neuroergonomics Approach. So I'm gonna be talking about something that is so intrinsic, but sometimes really oftentimes really overlooked, which are environments. So let me start by saying that we have an innate connection with nature. When we think about natural environments, we often associate them with their beauty in terms of they can grab our attention effortlessly. We can sense a feeling of escape where we look them to just go from our daily lives. They have this fascination and expansiveness where we can have this freedom in those environments and we actually feel pleasant being them. As compared to more built or urban environments, which are oftentimes overstimulating. There are many things going on the streets. Sometimes we feel some sense of confinement, having these high skyscrapers and just feeling just narrow streets. And so we feel this lack of escape or limited self-fascination. And considering that actually it's by 2050, it's expected that more than 2/3 of the population will be living in urban or city environments, and also that we approximately spend 90% of our time indoors, we are going away or shifting our attention from these natural environments, which oftentimes present a lot of benefits to ourselves and our overall well-being. So with that being said, I want to show some of the benefits that nature has, such as the power of restorativeness. So according to the attention restoration theory, it proposes a model in which it explains that natural environments can actually help us improve cognitive performance. So when we're doing any task at the beginning, we have our attention and our focus to that task, and that's what they call directed attention. So that's going well, but after a certain point of time, you are experiencing some fatigue. You are already like tired of writing that grand proposal or anything. So you need that break from your day-to-day to really restore those energy levels. So that's where this theory comes, and it says that through experiencing nature, you can have this involuntary attention and gain this restored attention that was lost at the beginning. So we can see many of these benefits in terms of attentional control, cognitive inhibition, working memory, and actually cognitive flexibility, in particularly in the prefrontal cortex, which is an area known for high executive order. Following up, we also know that nature has a role in stress recovery. According to this other theory, the stress recovery theory, it proposes also a framework showing that natural environments can contribute to stress reduction. So by being in natural environments, we oftentimes, not only in our brain or our attention, but also we feel it in our body. We feel more relaxed in these sort of environments as compared to other urban environments where there's a lot of things happening, lights, people walking, bikes, traffic, etc. So this theory also proposes that by being in natural environments, we can actually improve our heart rate variability, with skin conductance response in terms of our parasympathetic activity. We can lower our blood pressure in terms of what it encompasses its physiology. So, but we are moving more to a digitalized world, right? We have our phones and especially virtual reality. So can we experience the same feelings, the same benefits of nature in virtual reality? So our lab had this question: can technology actually bridge the gap between our needs of nature and our increasingly digital lives? And for that reason, we decided to explore the physiological responses, psychophysiological responses in real and virtual world environments. So by exploring, we want to investigate real-world and virtual reality environments in terms of their impact on neural and physiological responses during rest, using a multimodal neuroergonomics approach, using mobile neuroimaging to monitor the brain, as well as wearable devices to monitor the body. So for our experiment design, we conducted let's say two phases of this experiment, which is a real-world immersion, as well as a virtual reality immersion. So for this, the participants were sitting at a resting state, and they were tasked with immerse and observe the environment for 5 minutes. They were presented 5 environments in a counterbalance sequence. This was also made in collaboration with Longwood Gardens, a botanical garden in Philadelphia. So we took our participants to the botanical garden where we did our recordings. And also we did a digital twin of the same environments using Unreal Engine. And after each environment of each exposure, we were presented some self-reported evaluation about their perception in terms of how restorative that environment was, or how they consider beauty in terms of nature or art. So in terms of our locations, we have 5 different locations. These are the names. And we kind of group them into what they have in common in terms of being built. That means more like a more manipulated environment, and natural environments, as well as indoors and outdoor environments, as well as individual environments, because they offer some unique, great perspective and great characteristics on their own. So we presented this along with Gardens, taking them as well in the VR world using Unreal Engine. So we're using this multimodal approach where we are using wearable and mobile monitoring. So for both, we're using FNIRS to capture brain activity. We're using the Empatica E4, which is a wearable wristband, to capture body in terms of electrodermal activity, temperature, motion, cardiac activity, as well as self-reported surveys to capture their subjective understanding of what's happening in their surroundings. As well as for the VR immersion part, we also had the eye tracking. So let's go to our preliminary results. And in terms of cognitive engagement between real world and virtual reality, what we're observing here in this graph is a factor from our engagement with beauty scales Sorby presented, in which what we're observing is as the individual starts perceiving that environment more aesthetically pleasing, we're observing an increase in terms of HBO levels for the right lateral prefrontal cortex for the real world environments as compared to the virtual reality. So this actually may reflect some attention regulation and more engaging of cognitive processing in terms of aesthetics, key to the restoration in art that I presented. This is also observed in this graph where we're just showing the levels of HBO mean, in which we're again observing this higher activity for the real world as compared for the virtual reality. And when comparing again built against natural, we're observing the same pattern where real world have higher activity as compared to the virtual reality. So whether they are in built or natural environments, we're observing more cognitive engagement in real world that might require greater cognitive processing for the prefrontal regions. And VR reduced sensory may lead to this lower engagement of prefrontal areas. Moving on in terms of physiology, we also capture these arousal and stress biomarkers. And what we're observing here is just the number of skin conductance responses in comparison of real world against virtual reality. And we are observing higher levels of arousal in virtual reality as compared for the real world. We're observing the same in terms of indoors and outdoors, so the same pattern in terms of VR being higher in terms of these arousal levels, particularly in the outdoor levels. So we are seeing this increase in physiological arousal that then tend to say that these VR environments tend to be more stimulating or even stressful at some points. And so in this other graph, what we're showing is also a graph showing another factor from our surveys, saying that as the individual was feeling more in terms of their body or sensations, more in relation to that environment, there is a higher level of skin conductance response for virtual reality and compared to the real world when we are seeing actually a decrease of this kind of like arousal. So in terms of real world, we are having a better support of stress recovery, aligning with this stress reduction theory. And actually, appreciation of beauty can help us lead to this calming effect, which is what we're observing in terms of these results. Moving on for our cardiac activity, again, comparing both realities, we are observing here that as well as the individual was perceiving the environment more aesthetically pleasing, we are seeing an increasing heart rate, particularly for the VR section. And we are seeing here also, compared throughout the 5 environments that were shown, the same pattern is shown, where the heart rate is actually higher for the VR when compared to the real world. This higher heart rate might indicate a higher level of physiological arousal and can be presented as a precursor maybe to stress reduction. As well as virtual realities might not be intended to imitate ideally or identically this real-world nature. However, they provide some elements of this natural beauty that can help achieve this stress recovery. And so overall, the lessons learned about this work, particularly in terms of going outside to the real world, in terms of mobile imaging, is to really remember to charge your devices before going out to conduct your experiments. Pack everything and test your equipment beforehand. I think there are many challenges that we are presenting, especially in many of the works that we're doing going outside from the lab, and I think that's really important to keep in mind. And in terms about these realities, is that they can offer different paths to restoration. We are seeing distinct psychophysiological responses due to these environmental factors. We are seeing higher arousal levels in virtual reality and higher cognitive engagement in terms of real world. So we're still trying to see more in-depth analysis of what's coming up or what are these biomarkers actually telling us. And so our key findings so far as well is, exposure to diverse environments is really crucial to have this optimal physiological and cognitive function. And VR might offer a potential personalized restoration intervention. And as I mentioned, there's future analysis still ongoing. I'm excited to see what's coming up next. And really compare these virtual and real world worlds, I think might have a lot of benefit for potential therapies or interventions moving along. Last but not least, I want to acknowledge my team in the Neuroeconomics and Neuroengineering Lab, the School of Drexel University for having this great opportunity, as well as Longwood Gardens for making this collaboration, going to this museum. So I really appreciate them. And without any further, I want to thank you. And if you have any questions, I'm here. Thank you.
Thank you, Kevin. Any questions? Please.
Did you think about a sham condition outdoors where the equipment burden was the same as the indoor? I mean, wearing that thing,
With the VR, you mean?
Yeah. Mm-hmm.
So can you rephrase your question just one more time?
Did you think about having a sham condition? Because you might see some effects of just the equipment itself, right? So
Okay.
sitting quietly in a garden. as opposed to sitting in a garden with the virtual reality headset on.
Yeah. I mean, of course, when we were like trying to do these studies, we have a lot of, let's say, noise coming out from going outside to the Longwood Gardens, and we would have just the light coming up in terms of higher in ambient light, or even the VR. With the VR, the eye tracking having some types of noise in terms of our brain signals. So we just tried to make sure that all of these confounding factors that we were finding through our testing procedures were the less as possible. And I think we just tried to make sure that our equipment was working properly, that we were not having any external factors in terms of signal messing with the data. So I think that's what we were just trying to do.
Hi, really interesting. Thank you so much. I had a question. A few times during your talk, you mentioned the beauty of the outdoors and how that might be linked to why we experience, or why the data were showing, why the data was specific to outdoors, being kind of linked to aesthetic pleasure and beauty. Did you consider or research, do you have any ideas about if the interior of the design, the building that you're in, if that's aesthetically pleasing? Buildings like this, I just am so much happier in them. And how did you really define beauty? Is there something that's more specific to each individual, where you really like a mountainscape, or you really like the beach? Some people are going to really respond to different aesthetics. Yeah,
I think for the first question, I think it's a really great question, because not all buildings are the same, or they don't have the same characteristics. So I would say that yes, there might be a difference in terms of how you perceive this environment, in terms to an office or something that you already think something else about that environment. So that's a great question to continue working and continue studying. So I think that's a really interesting work, continue moving forward. And in terms of the beauty, as you said, it's really subjective to person. We try to capture as more of a generalized idea based on our surveys that we presented to the individuals. And in those questions, we were just asking them, what do you think about nature in terms of like a rating scale, and see how they would perceive. I can actually, after the talk or something, give you more about the surveys that we used, which one is called Engagement with Beauty Scale, and that's kind of 14 questions mainly about how they perceive beauty in terms of nature or art, or even morally or spirituality and so on. But yeah, we have a little bit of a list of what we're using beauty.
Okay, great. Thank you so much.
No problem.
Thank you again, Kevin. Our next speaker is Emily Waskow. Welcome, Emily.
Hi, everyone. My name is Emily Waskow. I'm the lab manager at the Center for Translational Cognitive Neuroscience over at the VA, and I work with Dr. Andrew Budson and Dr. Catherine Turk there. Thank you so much for having me. So I wanted to begin by quickly introducing everybody to Alzheimer's disease. As we know, Alzheimer's is a neurodegenerative disorder, and it gradually destroys one's ability to learn, reason, and carry out daily activities. And as our population is aging and Alzheimer's disease becomes increasingly common, we need to evaluate the efficacy of treatments, specifically medications, in Alzheimer's, and how they interact with complex processes, specifically sleep and episodic memory, and the bidirectional relationship between them. I also wanted to define mild cognitive impairment and mild dementia. So mild cognitive impairment can be due to a variety of disorders. In this talk, I'll be mentioning it specifically for Alzheimer's disease. And mild cognitive impairment is when there's no deficit in functional activities, but we're still seeing a cognitive deficit. And mild dementia is when we begin to see a deficit in instrumental activities, such as managing your finances, cooking, transportation, but we're not looking at moderate to severe. So these people are still fully capable to, go to the bathroom by themselves, all of that. So as everybody has already defined, we were looking at event-related potentials. On here you can see that ERPs distinguish mild Alzheimer's disease from healthy controls. Specifically, ERPs are time-locked to tasks and non-invasive, which is different from many other ways we can define Alzheimer's. And we're looking at the study specifically at the LPC, the late positive component, which we observe 500 to 800 milliseconds after a stimulus. And that is directly linked to cognitive processes like memory. So in Alzheimer's disease, sleep disruption is an early and consistent correlate of mild cognitive impairment and mild Alzheimer's disease dementia. And examining behavioral and ERP measures of memory can reveal electrophysiological and performance-related changes, which can help us understand the relationship between sleep and cognitive decline. In this study, we aim to establish whether wearable technologies can generate data that's sufficiently reproducible and sensitive to track and interpret changes in memory after 2 specific interventions. So one is after approximately 12 hours of rest. So this would be from 5 to 10 a.m., and then the other one is after around 12 hours of daily activities, so from 5 to 10 p.m. We recently concluded our data collection, so just to preface this, our analyses are not fully complete, but I'm happy to share the data we have now. So we were using these 2 wearables. One, we were hoping to detect the level of daytime and activity and sleep characteristics using an Empatica Embrace Plus watch. We were hoping to replicate reliable electrophysiological markers of recognition memory using a dry EEG headset from Cumulus Neuroscience and a corresponding tablet. And we were looking at memory changes after sleep and wakefulness. We also wanted to establish the practicality of using these wearables in a cognitively impaired population. So we hypothesized that wearables would be feasible in a cognitively impaired population, and we expected that memory enhancement would occur during sleep as we can track using the Cumulus headset. We had 37 total participants, 12 of which were younger adults, 13 of which were older adults, and we had 12 older adults with mild cognitive impairment or mild Alzheimer's disease dementia, and I'll just be referring to them as MCI patients to continue. Our average young adult age was 23 years old. Our older adults were around 73 years old, and they did not significantly differ in age from our mild cognitive impairment participants, and all of the groups were highly educated with around a bachelor's degree, and the groups did not differ in years of education. So we measured sleep through the Embrace Plus actigraphy tracker. Patients linked that to their smartphones, and they updated it around every day just to make sure everything was synced properly. They also completed daily sleep diaries, asking about how long they slept, their quality of sleep, and some activities they did or did not complete during the day, such as taking a nap, exercising, having any caffeinated beverages. And after the first week, which they just onboarded with the Empatica watch and got accustomed to the sleep diaries, they took a survey of their sleep quality, and after the ERP paradigm, they completed that same survey again. We measured ERPs through a dry EEG headset from Cumulus Neuroscience. So at the beginning of the study, they came into the lab and we fit them to the right headset. So we measured their head and we taught them how to put on the headset themselves. As you can see on the top left, there are little sticky electrodes which go behind the participants' ears on the mastoid, and then the participants were responsible for putting on the EEG headset themselves and linking it to the tablet. There were instructional materials that came with the headset, and on the tablet, you can see it walked you through some of the process and gave general reminders such as not to eat or drink while you were wearing the headset, not to move around too much, etc. And then it took you through a signal check so that you could see, make sure that each of the electrodes had proper connection before you continued. We used a continuous recognition paradigm involving images that were new and that they had previously seen. The previously seen images were either presented in the previous session or across multiple sessions. So in each session, we presented them with 100 images, 40 of which were brand new, 40 of which they had only seen in the session before, and 20 of which had been presented in all the previous sessions in that block. We'll go more into the schedule in just a second. And this task was made into a game. So it was a rogue agent task, and the subjects had to determine if the image was brand new, they'd never seen it before, or if it was a copy image, they had already seen it. So we had 2 counterbalance conditions for the study. In the first one, the participants began their session in the morning, and then for the second block, that second week, they started in the evening, and vice versa with the second counterbalance. As you can see, for days 1 through 5, for counterbalance 1, they were completing our standard primary paradigm, which was about 3 minutes of resting state EEG, and then the memory paradigm that we just discussed. And for the last session, the 11th session in each block, they completed resting state, a mismatch negativity paradigm, and visual oddball, and they only completed that one twice. The sessions usually took about 15 minutes, and it was every morning and every evening. We wanted to make sure it was doable, and then that last session was about 45 minutes. So we had over 700 total sessions, and we were really pleased with our ERP adherence results. The average adherence to the protocol was high, even for cognitively impaired participants, and there was no significant difference between groups in terms of adherence. For our actigraphy data, we were able to see the amount of movement, skin conductance, their pulse rate variability, and skin temperature. And we could see that for the full 21 days, so the first 7 days of the acclimation and the 14 days in which they're also doing the ERP paradigm. And it looks like the data is adequate to conduct in our intended future analyses, such as examining the differences between the subjective and objective sleep data, so whether or not the participants were accurate when they were doing their diaries, if the time they thought they were sleeping and the number of times they woke up, if that was consistent with what we saw objectively from the watch. We are also interested in investigating the impact of sleep time and quality on memory, so from both their behavioral responses from the task and their ERP responses. And our ERP data quality results also looks pretty good. In this figure, you can see the red line is a novel image, so if it was the first time they ever saw it. Orange was images they had seen once before, and green is images they've seen multiple times. The blue line is an average of the green and the orange line, so just noting that they had been seen before. And this isn't showing us any differences between groups. This is just a preliminary grand average, so it's grand averaged by users and across users, but we can see some changes, which is exciting. For our future analyses, we want to test that our ERPs have sufficient sensitivity, reliability, and practicality to detect these memory changes. As I mentioned, comparing the objective sleep results to the subjective, and comparing our 3 groups to confirm known effects of age and disease on sleep and memory consolidation. We're also excited to explore potential memory changes that occur during sleep between groups by looking at the LPC component across AM and PM sessions. So far what we found is that the patients with mild cognitive impairment and mild AD dementia were able to adhere to a complicated at-home study design with wearables, and that these wearables may be employed in at-home paradigms for screening, staging, and longitudinal monitoring of brain function. We're still planning to test our data for consistency with past observations regarding age, Alzheimer's disease, and sleep on memory consolidation. And the implications of this are huge. This could potentially be used for medical trials. So for drug development, once patients are administered a new drug, they can take these headsets home and we can track their electrophysiological changes over time. So patients with cognitive impairment aren't always the most reliable, comprehensive, and accurate historians of how their memory is. And this will just give us another means to test how they're doing, if they're having any effects of the medication. I'd like to thank my wonderful team at the Center for Translational Cognitive Neuroscience and some of our study partners for our funding support from Bristol Myers Squibb Cumulus Neuroscience and Empatica for our wearables, and most importantly, our wonderful study participants from both the VA and the BU Alzheimer's Disease Research Center. And thank you guys so much for inviting me to speak today. I really appreciate it.
Thank you very much, Emily. Do we have any questions? Yes.
Yeah, wow.
Yeah.
That's impressive. You make it sound so easy.
It definitely had its challenges, but it was fun.
Yeah, what were just some of the challenges in getting here?
Right, so Wi-Fi connection was a big one. Participants in general were responsible to connect the tablet to their Wi-Fi at home, which could be difficult. We spent a lot of time being tech support. It was an interesting experience, and just helping participants with connection. So I had mentioned the little adhesive electrodes, and some patients had trouble either putting those on with their nails, with the hand mobility, and sometimes they had to snap in the cables. Sometimes they just had a hard time snapping those in. So we worked with patients to adjust as needed. We had a participant who would snap them in beforehand and then attach it. So it was just being flexible, being open-minded, and really taking the time to work with each participant to make sure it worked.
Yeah, this is really cool. I sort of had a related question of, did you, for the patients, have any protocols for care partner instructions, or was it just for the patients themselves?
Yeah, that's a good question. Some people had caregivers that would help them. Many people didn't. So if they had a care partner that helped them with transportation especially, and they could be there during the initial sessions, they also watched how to put on the headset, connect with the tablet, all of that. If not, the instructions on the tablet were pretty comprehensive for help putting it on. So patients were generally able to do it by themselves, and we really encouraged independence, but caregivers definitely played a role for some patients.
My second, just quick question was so did you notice individual difference with alignment? I don't know if that sort of could play a role, or how you controlled for that at all?
Yeah, so we had patients, some of them stand in front of a mirror when they put it on. There was a little tick mark that they aimed to put right in the middle, and that helped them with alignment. And we made sure that we had them practice in front of us in the lab, putting it on to help them with the fit as best as possible. There were some alignment issues, but we're hoping that those will kind of average themselves out.
Thank you. This was awesome.
Yeah.
Thank you very much. Okay, we made it to our last speaker, Dalin Yang. Welcome.
All right, thank you for introduction. And my name is Dalin Yang, and from a postdoc from Washington University School of Medicine. Can you go? Thanks for the organizer to have me and present our work. Autism spectrum disorder is a prevalent neurodevelopmental condition. In the United States, around 1 in 36 children will have impacted by this disease. And more than 70% of the autism have at least one co-occurrent psychiatric or neurological brain disease, such like brain seizure, anxiety, and ADHD. The core symptom of ASD, including the persistent deficits in the social communication and the enhanced restrictive behaviors. Most autism are diagnosed after 4 years old, even though the studies show that the autism can be validated around 24 months old. Also, early diagnosis will facilitate the early intervention and further improve the life quality of the patients. Therefore, the phenotypic biomarkers is needed for this field. Motor and imitation difference may present the early phenotypic biomarker, as the imitation is crucial and insufficient part of the early social skill learning, like social bonding and the language development. Motor imitation has been shown to be impacted by in the autistic children. Specifically, current studies show that motor imitation deficits seen in the ASD may be related to the underlying visual motor integration deficits, because the children with the ASD more rely on their proprioceptive inputs rather than their visual inputs. Currently, they have a lot of the motor imitation assessment approach. They want to use those approach to do the motor deficit detection. Mostly is a behavior-based assessment. The one important one is called the human observation coding. It's shown in the right table. It's quite time-consuming, also required extensive training. It's practical for the clinical and the home setting. And they also have the computer vision-based assessment, and they are kind of the categorized metric, so they just give you the index either that is good or bad. Also capture mostly like the large variance, like hand wave or the hand push-up. So in conclusion, the current approach about the motor imitation assessment is limited in their ability to the efficiently detect the wider variability in behavior presentation and the heterogeneity in the human motion between the autistic and the neurotypical control. Our team member in the Joe Homskin and the KKI, they developed a new computer vision method that is called automatic computational assessment of the motor imitation. Basically, they use a dynamic time wiping method and a metric learning method to integrate the spatial and the temporal difference during the movements, and can require don't require the extensive human inputs and code training, and can provide a more generalized and objective and continuous score of the imaging fidelity. So they also compare the CAMI score with the currently widely used human observation coding method. Is very highly correlated with each other. Also is outperform than the HOC method. So the open question here is, what's the underlying neural mechanism of the motor imitation? So it's still unclear. So people said that the mirror neural system is always engaged when one performs the motor action or when one views the other people performing action, implicating the social cognition, language, or series of mind. So that said, mirror neural systems support observation learning and promote imitation. Those systems mostly including the premotor cortex, inferior parietal lobe, and the inferior frontal gyrus, and the superior temporal suckers. They also found that the differential mirror neural system activation has been shown during the action observation and the imitation of the small finger movement or facial expression. Currently, I think they have a lot of a challenge using current neural imaging modality for autism field, specifically when we want to study the motor imitation task. Our gold standard imaging modality like FMR is relevant to have a loud noise and have a confined space, mostly impossible to do the dancing movement inside of the FMR scanner. The optical imaging is kind of quiet and open scanning, provide open scanning and a more naturalistic environment, even though previous study used the SPICE FNIRS array to do the autism-related study, but that is have the low anatomical specificity. Provide a high density because we have the overlapping array, and we can go into the deeper brain tissue, also can have the higher anatomical specificity. The other potential impact is the motion artifact can potentially degrade our image quality and the data reliability. So optical imaging is relatively less sensitive to the motion artifact compared to the fMRI and EEG. Also, current motion sensory algorithm can help to further improve the image quality. So the aim of our study, eventually we wanted to precisely establish the brain-behavior association underlying of the imitation in autistic school age and preschool children. But in this current study, we aim to validate the feasibility of the motor imitation paradigm and establish the brain behavior association in autistic and non-autistic adults and school age children. So we hypothesize that first, the motor imitation paradigm is feasible in autistic and non-autistic adults and school-age children. Secondly, observation and imitation will engage viral motor brain area. Region in the mirror neural system will be activated when observing or imitating the motor movements. Lastly, we also hypothesize that brain activity in the motor region will positively correlated with the imitation fidelity. Also, the brain activity in the mirror neural system negatively correlated with the autistic traits, like SR score. Thanks to introduce the HD duty in this morning. So these slides, I'm going to briefly introduce what is HD duty. HDUT is also the optical imaging modality that uses near-infrared light to measure the tissue oxygenation, resulting in the hemodynamic measurements of the neural activity. Compared with the other NIAS device, HDUT demonstrates the capability for mapping the human brain function with image quality comparable to fMR. Our simulation study showed that the current localization error is less than 5 millimeters. So this right figure is show our home-developed HD-DOC system. It has 128 source and 125 detector. We can have more than 3,000 measurements with the source detector separation less than 40 millimeter. And the sampling rate is 10 Hertz. The penetration depth is up to 25 millimeter below to the scope surface. We also overlap with the currently widely used brain pixelation, golden pixelation. We can cover the visual, temporal, parietal, some prefrontal area, totally including 10 functional networks. So this is an ongoing project. We already collect data from 184 participants, 103 is adults, and the 81 school children. Beside of the HD-DOT data, we also collect the computerized assessment of the motor imitation, that's called CAMI score. We also calculate the social responsiveness scale, is kind of the relative the autistic severity, and also record the Edinburgh Happiness Inventory and the Kaufman Brief Intelligent Test. So the right figure shows our demographic information, but in this presentation, I will mainly discuss the result in the adults cohorts. So this figure shows the CAMI score. So the score is near 1 as indicated the participant performed the motor movement well. And you can see the NAC, neurotypical control, have a higher CAMI score compared to ASD. In terms of the severity, so ASD always have a higher SRS score compared to the NAC, no matter in the raw score and the T-score. For the IQ score, among those three groups, we have the similar IQ score. For our HD-DOT scan protocol, we first ask participants to do 4 different functional localizer tasks. First of is auditory. We play the hearing words task, and the other one is a motor task, is finger typing. And we also have the visual checkboard task and a movie watching task. For the imitation and the observation task, we have each task have 6 blocks. Each task period is 18 to 21 seconds. For the observation task, we ask a participant as stay as possible and watching this avatar do the movements. And for the imitation case, we also try to keep their head don't move and then mimic this movement from the avatar. This is our data analysis pipeline. We do the header modeling, light modeling, pre-processing, and risk construction and spectroscopy. I'm not going to introduce the details, but all the script and tutorials is available from NITIC and GitHub. Because this is a motor-related task, we want to detect and sensory the motion. We use the global variance of the temporal derivative method. It's called JVTD. Basically, we are going to detect global instantaneous change in the optical time trees. This method is inspired by the device. It's shown in the right figure. So you see the grid plot. We have the vertical line. That's the area is indicated the motion. So it's responding to the red time trees in the above. So similarly, our JVTD also can detect those motion, and we can use its JVTD time trees to sensory the motion period and remove them from the further analysis. This slide is going to show our localized mean map. The purpose is going to establish the fidelity of our system. So when we play the audio task, you can see the bilateral auditory area have the higher activation. Similarly, for the visual task, we can see when we control lateral activation in each hemisphere. This is for the motor task, have a similar activation. This is for our main results about the motor observation and imitation task. In this case, we use a general linear model to calculate the beta value of the response of each task relative to rest. So the motor observation task in the left panel is show when we participant watching the avatar do the movement, they have the visual area and the social area have the activation. Come to the imitation, because the motor action involved, so we also see the activation in the motor area. And besides that, we also do the PESO BC analysis because the PESO due to use the functional defined golden pencilation provide a framework to couch our brain behavior relationship within the functional defined region of interest. The result is quite consistent with our Waskow BC analysis. After we get the PESO BC, the beta value, we also calculate the brain behavior association. This slide is going to show the brain behavior association between beta value and the SRT score. So we found that in the motor observation case, we found the right temporal parietal junction, TPG area, and the right superior temporal gyrus area have a significant negative correlation. That means the participant have more severe symptom, have the lower activation in this area. Interestingly, we also see in the motor imitation case, the TPG area have the negative correlation, but it's happening in the left hemisphere. It's kind of the lateralization effect happen here. Come to our brain behavior association between the beta and the CHEMI score. So we found that in the motor area, we have the positive correlation. That means the participant have the good motor performance, they have the higher activation in the motor area. Also, we found the brain area bilateral inferior frontal gyrus, IFG, and the middle temporal gyrus, MTG, have the negative correlation. In summary, in this study, we established the feasibility using HD-DUT to measure the brain function during the motor imitation paradigm in autistic adults and non-autistic adults. And the JVTD ensure the HD-DUT fidelity and the sensory the motion, and enable us to interrogate the brain function related to the imitation and the observation. Also, HD-DUT-driven brain map implicated the region in the mirror neural system during the both motor imitation and observation task. Our brain activity in the mirror neural system negatively correlated with the autistic traits. Also, we found the mirror neural system have the motor area also have the positive correlation with our imitation fidelity. We also have the ongoing work, and we are going to apply this paradigm on our school age and preschool age children. And also, recently our study showing that the autistic children have the eye gaze difference. So we wanted to further investigate how simultaneous Aggies and brain function contribute to the disrupted brain mechanism related to the motor visual integration. And also, like what Joe said in the morning, we have a bunch of the data for the preschool children without ASD likelihood. We wanted to do the semantic decoding for this project. In the end, I would like to appreciate all my co-authors, mentors, and lab mates for their efforts for this work, and all my participants contributed their data. Thank you for your attention.
Thank you, Dalin. Do we have any questions? Yes, please.
Thank you for your beautiful presentation.
Thank
I
you.
really appreciate it. I would like you to comment just a little bit about the desensitization protocol for the children autism group, please. You know, the desensitization before doing the measurements.
You mean the data registration?
The preparation and the instructions for the children, so they just not keep, you know, with the hands in the sensors or moving too much.
Oh, yeah. For this result, I mainly showing for the adults participant data. Yeah, definitely for the kids. We try to avoid to tell them don't move, but you know, some preschool kids is they won't listen you. Yeah, so that is the challenge part for the toddler study. Yeah, but yes, thank you.
Any other question, either for Dalin or the previous speakers? No? Okay. So in that case, we'll have like a 7 minutes break. We'll come back at 4 for the student wrap-up.
Let me just introduce the session, and then I'll hand it over. So thank you all for staying until the very end. We have one more. Who's now? As I said at the beginning of the conference, we are really fortunate to have the Chen Institute sponsor this meeting. And because they sponsor this meeting, we're able to sponsor students to travel from all over the world to present their work today. So we've handed the task over to Adrian and Xian. They're going to try to address 2 questions we posed to them as part of the conference, and they can address the questions and the answers. But we're hoping that you'll also get to introduce yourselves and tell us how far you've come from and what area you're in. So I'm going to hand it over to these guys. I think the general idea is you guys get to talk a lot, so we'll listen. And if we run out of things to talk about, we'll ask questions, but it's really the floor is for you folks.
Okay, thanks a lot, first of all, on behalf of the Trainee Travel Award winners. And I would just go ahead and introduce myself. So my name is Sein Jeung, and I'm a doctoral candidate based in Berlin. And I'm using mobile EEG to investigate spatial navigation.
I'm also going to thank the committee for the Travel Awards and the Institute for providing the funding to make this whole thing possible, and of course the organizers of the new conference. My name is Adrian Kurt. I'm a postdoctoral fellow at Drexel University, and I'm a sort of a neuroergonomics generalist, but mostly FNIRS, and here also for navigation as well.
Hello, everyone. I'm John McLinden. I'm a PhD candidate from the University of Rhode Island, coming from Rhode Island, and my area of interest and research is multimodal neuroimaging, EEG, and FNIRS.
Hi, everyone. My name is Candida Barreto. I'm a postdoc at Drexel, and my research is on pediatric neuroimaging, especially using FNIRS on clinical and healthy populations.
Hi, everyone. My name is Dalin Yang. It's from Washington University School of Medicine. My main research is about the brain mechanism decoding using the multi-neuroimaging modality as along with the computer vision and the machine learning algorithm. Thank you.
Well, thank you for the awards and for having us here. My name is Kevin Ramirez. I am a PhD candidate at Drexel University, and my work is more about nature appreciation in terms of real and virtual environments.
Hi everyone, thank you so much to the organisers for the opportunity. My name is Layla Kawara. I'm from the University of Leeds. I'm affiliated with the Immersive Cognition team and also the longitudinal cohort study born in Bradford. My research is looking at adolescent brain development, and I use EEG in the classroom.
Hi, I'm Lara Papin. I want to thank you all for the opportunity for us to be here and also speak here. I am from Germany, from the north of Germany, from the University of Oldenburg, which is a place probably most of you don't know. But it's very close to the sea. It's a very cute little town. And yeah, my research mainly focuses on cognitive motor dual tasking and assessing it using mobile EEG in mainly real-world naturalistic settings.
Hi everyone, my name is Sol Morrissey. I'm from the University of East Anglia in the east of England. Thank you so much for the organisers for the award and for the invitation. It's been a great conference. I've really enjoyed everyone's talks. My research is in dementia research. Specifically, I look at how do cognitive changes in aging and early dementia relate to driving behaviour. So thank you so much. It's been a great conference.
Hi everyone, my name is Sabrina von Au. I'm from the German Sport University, and I will say thank you to everyone that we could be here, because without the award I couldn't be here, so thank you a lot. My research is on nonverbal behavior, in particular about self-touch, pleasant self-touch, and FNIRS.
Hello everyone, my name is Chun Young-shin from South Korea, and I've just started my PhD in Ewha Women's University, also in South Korea. And I was part of the research developing AI device that detect mild cognitive impairments in older adults. And specifically, my role was to extracting linguistic features which can differentiate MCI from healthy controls. Thank you.
Yeah, hello, I'm Torge Haupt. I'm also from the University of Oldenburg as Lara. I'm working on auditory perception in daily life, so we want to send participants into their daily natural behavior life without controlling anything, and want to basically understand the neural underpinnings of auditory perception. And yeah, thank you so much for the award. Otherwise, I also wouldn't have been able to attend this wonderful conference.
Hi, I'm Condell Eastman from Rensselaer Polytechnic Institute over in Albany, or upstate New York, so close to Albany. And I just want to extend one last thank you to the Chen Institute, as well as everybody who made this conference happen for the second year in a row. Thank you for the incredible conference. Oh, and my research is mostly focused on applications of deep learning to FNIRS data.
Okay, thank you everyone for the round of introduction. A hallmark of a good conference is when it leads to tangible changes and impacts in the way we think about our research and way we do our research. So we would like to share what we have learned in the past 2 days and what has been most inspiring, and how it would affect the way we approach our own works or even future works. So if I may make a point first, it really made me realize how important it is for studies that are done in naturalistic setups to share the protocol and potentially the data, because as the recording gets more and more multimodal and more complex, it becomes harder to reproduce or replicate these results. So we have seen many studies that have lower sample size, and I think one remedy is to help people interpret the data and also do meta-analysis by sharing the protocols more. So that would contribute to the longevity of the data sets as well as the general health of the field. Yeah.
So I think the original question was also with the what have you learned from the conference and how it applies generally, and I agree about the sharing of the methodologies and the data. It's definitely a very useful thing. My personal thing here, I think, is a little bit that it's sort of like, you know, from that perspective, it's a little bit like neuroscience versus the everyday world, because we have to sort of get over all the different hurdles. With my research that I presented for one of the posters, it was also based on you know, being able to walk with FNIRs, and there's a lot of different struggles with having you know, unconstrained participants moving around in mobile settings and how to handle these different things. And it's also it's also difficult data to acquire. So I think that, you know, sort of the meeting of the minds there is a good way to handle that. I also am always excited about new methods and then trying to apply them, and they always like give me like sort of like a naive hope that this is the one that will fix everything, and then I dive into it. So that's always an exciting thing that you know, I'm not gonna be pessimistic about, but it's gonna work.
Yeah, I think one of the things that I was most struck by during this conference was really the richness of behavioral data and data that comes from non-neuroimaging modalities. I really think, at least from my background, we do tend to get caught up in signal processing and delving really deeply into neuroimaging, but there's a lot of complementary information that can be provided by non-neuroimaging modalities, that referring to like physiological signals from other sources or behavioral data, especially in these naturalistic settings. I think it's really been impressive on me about how much you can get from that. So thank you for opening my eyes to that.
Well, for me, I think the clinical applications that we saw here, like translating basic research to real applications, such as we saw, I think it was today, yeah, in the first talk in the morning. It was amazing. It creates me like inspiration to continue, because sometimes when we do neuroimaging, we finish like, okay, we found this activation, that activation, deactivation. And I was talking with my colleagues while we were coming here, like, so what? What is the next step of finding brain activation? What can we do with that in reality? And then here in the conference, I had the opportunity to see how people are applying this knowledge to move forward. And for me, it was the main point of the conference. And I'm coming home now with my brain burning and thinking about a lot of different research that, you know, new methods, new things that we can apply. So it was really inspiring for me, and it was really nice for me to be here today.
All right, I think these 2 days I keep thinking 2 things. The first thing is about between the naturalistic and the precision. So I feel in our team we try to minimize the size of our hardware, and we also want to keep the precision as much as possible and can get a solid result and comparable with FMR. So I feel this, I think, make the most challenging part to become naturalistic, you know, have a small device and also have the very high resolution stuff. I will keep thinking about this thing, and yeah, that is one thing I keep thinking. And the other thing is about the sample size. Because I have the passion about the AR and deep learning, machine learning stuff, but it's quite limited by the sample size. Specifically, every team have their own samples and use a different paradigm. So our team is developing the toolbox to can convert a different type of the data, and we also have the cloud-based space. So we encourage if anybody want to share their data, so we can you know, collect a lot of data in the cloud space and do the large-scale analysis. So I feel that's 2 things I learned from this conference and inspire me for my future study. Thank you.
Yeah, I think for me during this conference, I've seen a lot of great talks, a lot of great experiences in terms of how to pushing the frontier of this neuroscience of the everyday world. And I think for me, what I have really learned is, you know, like you see all of these great advances, great studies, but behind of those, there are like a lot of challenges and limitations. And so I think sharing those challenges and limitations and, you know, like just collaborating with others, just trying to pick the brain of everyone in terms of the equipment, the systems, the signal processing, I think that has been really, really, really helpful or like really beneficial that I want to go continue forward and try to be like as open as I possibly can in terms of what I know and the things that I don't know, the things that I have been struggling with and the things that, you know, I can learn from others so that I can actually apply them and have actual great quality questions and data. So I think that's something that I've been just trying to explore, especially for like all of these great experts around us. So I think that's really, really something that I want to take with me.
Yeah, I completely agree. I think that what we're doing is very novel and it's very brave. I think historically the focus has been very much on control and how we can control the environment. So taking neuroscience out of the lab and into these kind of uncontrolled and often unpredictable environments is very brave. So coming to a conference like this, I've been really racking people's brains, like what system did you use? What equipment did you use? How did you control motion artifacts? These kind of things are really going to help me in my personal work. As I said, I do EEG in the classroom, and there's not much research yet, so it's really exciting to be part of this shift and to learn from everyone at the conference on the approaches. And it's already been mentioned so far, but protocol sharing right now is so important. There doesn't seem to be that many guidelines and that many kind of playbooks on how we should go about taking neuroscience out of the constraints of the lab. But I imagine that that's going to be a really big part of research in the next few years, and especially the discussions that kind of come from this conference, I think will inform those research studies.
Yeah, I also agree with everything that has been said already. Yeah, as in every conference, it's always very interesting to talk to many people from very different backgrounds, and for me it's also very enriching for my research to see all of these different angles and to learn from all of these different angles, because sometimes even people who are not into like particular what you are doing have very novel ideas and share them with you, and sometimes when you're just with your colleagues you don't even think about these things, and yeah, it's really, really interesting to to hear from these angles. And I also have to say that in general everything has been very nice, and the people have been very nice and respectful, and that led to quite fruitful discussions. And yeah, thank you.
I think something which just came up in quite a few talks is the importance of multidisciplinary teams being integrated into real-world research, because there's so many challenges that comes up in managing these co-founding variables. And so for many of these talks that have came up, people have been working with commercial partners, engineering teams, geospatial teams, and I think this particularly real-world research outside of the lab requires so many different levels of expertise. And this is something that quite a few talks have touched upon, which I think I'll definitely be looking to take forward into my research, which has been really interesting.
I've been really inspired by what can be investigated, what is possible with neuroscience of the everyday world, and I have always this dream that I can investigate children and persons with severe multiple disabilities, and this is really challenging because you can control nothing. And that would be one dream, and I can follow this dream. And I learned here that I love science and I love my field, and at the same time I suffer a lot and feel lonely, and then I came here and see what can be happened and how we can solve together problems. And yeah, I like that a lot. Thank you.
At this conference, I've earned some valuable insights. One major takeaway for me is the importance of different data, different types of data, like linguistic input and number cues or biological things should be integrated in FNIR's device or to like improved prediction accuracy. So for me, I'm more familiar with dealing with only linguistic data, but from now on, after this conference, I should be more experienced, more FNIR's data. Yeah, thank you.
Yeah, for me, the summer has been already quite intense. I went to France to the Neuroergonomics conference, where I think a lot of European research groups met and discussed similar topics. And now to be here in Boston, to see what the American landscape has to offer was really, really interesting. And I think two major things I took from this conference. One is like the practical applications. So throughout all the different talks and seeing all these different posters, I was insanely impressed by how far this field has come in terms of actually producing something meaningful, and especially in clinical applications, to see how all this research has been fruitful and actually improved people's lives. And seeing this in terms of videos, that was really, really impressive to see. So I'm glad that years of research have built up to this, and we can experience actually this time where products are developed that help people, especially also the different companies. That the constant therapy talk, for example, I thought was super interesting. And the second major point that I learned, or that I was like reminded of, is that if you work with everyday world data, multimodal data, is that you have so many different angles that you can look at data. So the high dimensionality of data that you are sometimes missing if you look at lab data is, I think, one of the strengths actually of this field, is that you can run many different analyses, that you can look at very different factors. And I think this is the beauty of it, because the complex interplay can be investigated like this, which is maybe not possible if you only look at isolated lab activities.
Sorry. So for me, coming from, I guess, more of a software development perspective, one thing that really jumped out to me, or that I learned here, is the approach that a lot of other people had when it comes to developing their own software, since that was actually surprisingly prevalent amongst some of the posters here, and a couple of the talks it was mentioned as well. But one thing that I noticed was a lot of people were focused on, when it came to software development, how, like, not only how it would be used and the ease of use by the people who would use it, but also like building the software where people already would use it. For example, there's somebody who's building a toolbox based on EEG Lab, and one of the biggest reasons for using EEG Lab was that that's just the most popular software that was the most popular software at the time for EEG processing. So kind of taking that mentality into how I'm developing the software that I'm trying to release, that I think that's one of the most valuable things that I've learned here.
All right, thank you. The second question that we were trying to address as part of this panel is a sort of a forward-thinking one, and it's, of course, what is the future of neuroscience in the everyday world? And I think, personally, there's two parts to that, right? There's what is the future of neuroscience, and we all have our methods and this sort of stuff. We'll have a new technology in 5 years and 10 years, new analysis and all this, and also the way we interpret the data. And then the other half of it is really, how is the world changing? Because, probably we're looking at how people drive cars that we may not drive in the future, and how classrooms work in ways when we're also reconsidering how we're going to be teaching kids. And then, you know, we saw with Dr. Hassan's work in the beginning, kind of what we can achieve by potentially just having all these cameras recording everywhere, which is sort of a privacy nightmare in the 60s and somehow is acceptable now. But I'd like to kind of like popcorn this, so instead of going down the line, if anyone is like ready to answer or not ready to answer, we can start wherever, and yeah.
All right, since I already have a microphone. So as far as the future of neuroscience, one thing that I think has been kind of an overarching theme this year has been reproducibility, more so than I think I've seen, especially compared to last year and many other conferences. It was touched on multiple times here in this panel, even in the neurotechnology panel. A lot of people have been focused on producing not only producing results, but releasing the data sets. There's more publicly available data, at least for FNIRS. Multiple data sets have been made public this year, more so than I think any other year. There's been, people are making their software or their code more publicly accessible, and even the CEO of Kernel was discussing trying to become more transparent or open with how they do things there. So I think the future of neuroscience reproducibility is definitely something that we're going to see continue, or at least that I certainly hope will continue to be a major facet of that. And then to the second point of how that'll change with the world, we've already seen people taking advantage of the rich amount of data that you get from your smartwatch or your Fitbit, from having your cell phone on you. And I think we're going to continue to see that grow, especially as more of these devices develop, like Facebook's or Meta's Ray-Ban, and maybe if Google Glass ever makes a reappearance.
All right. Do you want to go? Yeah, yeah.
Yeah, so I'm on the spot now. So I think that what we have seen here already is when like moving from the lab to the real world, that we, instead of try to control everything in the environment, rather record what's going on in the environment. And I think this is very crucial. So we don't need to like not have this information at all by controlling it ourselves, but we can use methods to record what's going on in the environment. And I think this is really what the future is. And I also totally agree that it's probably very handy to use the devices that people have anyways, and that they use anyways, because this is how you can convince someone to participate in a study most easily. Yeah, and what I also see a lot, and what I also found very impressive here, was that so many different devices are integrated at the same time, which what I learned from my project so far is can be quite hard, especially in terms of timing and everything. So I think that this is going to become way easier in the future.
Just on the point that people were making there about the passive data collection of things like smartwatches, mobile phones, I think it's going to be really interesting in the future to see how that gets integrated amongst the clinic. Because at the moment, I think, so the majority of diagnostic work still takes place in the clinic, and that includes the neuropsychological tests for things like neurodegenerative diseases. But if we're able to integrate kind of these passive data sets that are continuously monitoring people's behaviors, and if we're able to have cognitive tests within people's homes, then we're able to measure this more frequently, and then clinicians will be able to use this information to make to help assist with the diagnostic process. So I think we're moving more into an approach which is preventative rather than just diagnostic on the spot, which is really important for an aging population in particular.
So one of the things that I think is going to change in the future, I'm going to switch topics a little bit here, but neuroimaging has been very heavily based on weird samples. So by that I mean Western, educated, industrialized, rich, democratic. And this is because we have very hard to reach populations. However, I do believe that taking neuroscience outside of the lab, it makes it much easier to reach these kind of populations. So in my work, I do EEG in the classroom. And I don't necessarily do that because I'm motivated by ecological validity. I'm not trying to replicate effects in the real world. I'm trying to widen my participant pool and diversify my participant pool, especially because I'm very interested in the effects of deprivation and poverty, and I can't really research that in the lab because we tend to get a specific type of demographic that volunteer to our studies. Whereas if we take neuroimaging, neuroscience into the everyday world, into people's everyday environments, then this is an excellent resource for us to gather these kind of participants. So for adolescents, for example, they are already in schools, and they are very rich and diverse areas for us to collect participants. And I believe that this is going to really change the scope of the research that we're collecting. Taking neuroscience into kind of homes, places of worship, schools, everyday environments is going to really resolve this issue that we have in that our demographics are very much focused on one type of person. And that's what I'm really excited about, because then we can serve the underserved populations that we care about and that we're trying to improve the lives of.
In my opinion, for the future, I feel in the neuroscience case, I feel so far the brain still is unclear, so we don't really understand what's going on inside the brain. But I'm really excited about the new technique like deep learning, machine learning algorithm, like ChatGPT is super powerful. So with those development of the technique, I feel in the future, the brain decoding will be, much better than currently what we know about the brain. And in terms of the hardware, I feel, in terms to make people use that, majority of people can use that, the size of the device should be small. Even we have the single neuron recording device. So I think the size, the future neuroimaging device will be small and portable and usable in the daily life. And the other future direction I feel is that, so I feel one of that is about bedside monitoring, brain monitoring. So far we don't have very good monitoring approach. Maybe with our future the development of the combined with multiple modality, and we can do better bedside monitoring. Similarly, for the clinical using regarding the mental health, you see in United States around 20% of the population have the mental health, different conditions. So maybe after we understanding the brain better, we can find the treatment, better treatment for those disorders.
Yeah, I think one thing that I think is very critical for the future of the field is to be very mindful of the term ecological validity. I think that often these neuroscience of everyday world papers pride themselves or argue for like having done something ecologically valid, where in my opinion taking a lab experiment out into the world and basically conducting a lab experiment outside in everyday life is not ecologically valid, because then the question is, what do we learn about the brain? And I think that seeing here all these different studies, I think this is one step that we are already taking, and I think that this is a crucial step to take, to be always mindful if the experimental paradigm or whatever we are doing is in the end ecologically valid. Can we claim that what we are doing is ecologically valid? Because I think this is a huge point one should consider when arguing for whatever we are doing is ecologically valid. I mean, looking back at the history of the lab of Oldenburg, I think we have very much done the same. We have conducted oddball paradigms outside in different contexts, Whether or not we learn something about the brain in everyday life is questionable, I would say. But nonetheless, I think this is a very critical point to take, and I think we have done so. But moving forward in the future, I think it is even more important to be mindful of not simply doing very simple lag experiment in different contexts. But I think this also adds to what Yuri's presentation was. Instead of what is not longer important is to generalize over different subjects, but in everyday life over different contexts. And I very much resonate with that statement. And yeah, this is just something to be mindful of, I think, which is very important, whether or not what we are doing is ecologically valid at the end of the day.
Looking toward the future of the neuroscience, applying these FNIRS devices is challenging as discussed the last year's conference. But now I'm thinking we can make a little progress when we design the FNIR device more personalized user interface. For example, in my experience, over the past year, I worked with nearly 300 older adults testing a neckband style hearing aid combined with ear EEG, and they are all over age 60. They are used to taking on and putting off the device, and we can collect the data from their comfortable setting, like clinical setting and also home-based setting. So in this time, we have to think about the device to be more user-friendly, and so we can applicate more naturalistic environments.
When it comes to the ecological validity or what kind of environments people find themselves in, I think the point becomes particularly interesting in the context of spatial navigation, because I feel like there is a shift in the definition of what is considered real space, real life space that is relevant for our everyday life. Some talks have already integrated this aspect by, say, using immersive virtual reality or looking at the interaction between the user and the digital platform. So the more time we spend in those virtual or artificial spaces that are not really physical space, it becomes more real. So in that sense, what we mean by ecologically valid, it changes in itself, like with the development of technology. So I've seen many studies argue that observations about navigation in lab settings don't generalize very well to real-life behavior, because participants are sitting in front of the desktop and then they're doing flat screen tasks. But ironically, we are actually spending more and more time looking at a flat screen and living in that space. And this will be even more confusing when say things like virtual reality, HMD will be more prevalent in entertainment industry. So I think that's an interesting thread to think about, and I'm very curious how this is going to unfold.
So I just want to make sure, in addition to all of you, if there's anybody, any student, any young person who wants to chime in, this is the opportunity. So we'll open it up once you're all done.
Yeah, one of the things that I think I'm really looking forward to in terms of neuroimaging in the everyday world is really kind of redefining what's possible and what's not possible, and the constraints of different neuroimaging techniques as both hardware and signal processing approaches improve. I think a lot of the work done here today is kind of challenged some of my preconceived notions about what's feasible, and I look forward to that being challenged even further in the future.
Well, I think for me, okay. So I think for me, in terms of what I envision in the future is, I see a lot of work that is being put on, and I think there are like many ideas that comes out of like new research. Like you open a can and there's like many different ways that you can follow up your same study. And I think it's just about opening and like knowing more stuff in terms of a lot of possibilities that you can just like take in terms of like the pathway. But I think like trying to really define what is like the main objective in terms of, as many of you guys have said, like limitation and constraints, and be like, I want to really focus on finding the answer, or trying to like find more about this area. I think, I don't know, something like that is really interesting for me.
We can open the floor to questions. If anyone else wants to add to the discussion of how the world will change, or maybe it won't.
I can add something. I think we all social, and I think that the future is that we investigate more interactions with a lot of different groups or in groups, not just a single person or a diet, to see how the natural behavior is in such groups. I think it's the future of neuroscience.
Yeah. I think that's an excellent point. I think we heard a lot of good topics here, starting with the reproducibility, which I think is a very reproducible finding in and of itself. We heard ecological validity, new devices, more devices that are already on us or will be on us soon, or in us, who knows. And I think the weird population part is also a very important thing, because as you know that college students between the age of 18 to 25 are the majority of the population according to neuroscience research. Yeah, so does anyone else have any kind of thing they want to add or ask? Yeah, please.
I think something that I'm really excited about going forward is using neuroscience, and especially these like wearable modalities for earlier and more accurate diagnosis, sort of when we're thinking about aging disorders, and also more targeted treatment. So being able to integrate them into the clinics, and being able to, when we're thinking about integrating behavioral interventions with neuromodulation and things like that, like really being able to enhance clinical outcomes and improve quality of life, things like that.
Yeah, I think that's an excellent point, especially when you can see like clinical impact in some sort of thing that actually gets to patients. Like that's always very rewarding.
I don't even know if I can reach this. One thing that I would just like to add is that I think there's a statistic out there that's probably really outdated at this point, but that it takes 17 years for something from the moment of discovery for it to be applied generally into clinical practice. And I think it'd be really great to get that number way down. So as we do more either mechanistic studies or applied studies, it'd be really wonderful to see collaborations with industry and collaborations with clinicians and kind of across disciplines to try to get the things that we're researching, the really important things, bring them to the people that really need them. So that would be one thing that I'd like to add.
Yeah. I also had an additional thing that popped up just now. What I would also be very interested in would be the effects of observation on participants, because I believe that when people feel observed, that they behave quite differently as opposed to not feeling observed or not being observed. And I think, this is probably also an implication on using devices people were anyways more, as I said earlier anyways, because using these data, we don't have situations where people feel constantly observed, which is very typical for the lab. and also for, I guess, all sorts of experiments that don't last a long time. And I know that there have been some experiments where experimenters then left the room and still recorded, but told the participants that now is not a situation where they are recorded, but it's still quite different because you're in a lab, and I mean, you probably still feel observed to a certain degree.
Anyone else? I don't.
It's actually not a related point, but I think it's quite important, and I do wonder, like, if it's kind of by definition possible at all for people to not be aware of being measured, but maybe the barrier is getting lower and lower with wearables becoming more commonplace, and yeah.
I mean, maybe if you acquire the data after it has been recorded anyways.
Yeah.
Like, for example, I'm wearing my smartwatch and I don't feel observed, but like if someone now asks me later on, may I have a look at your heart rate variability, which is recorded anyways, that could be a way, right?
The point that I wanted to bring up is I've been really curious about this during this entire conference. So I wonder if the conventional way of hypothesis testing or the way we do statistics actually applies to those super exploratory studies, and whether anyone has a good idea on an alternative way to do it. So we did hear that for lower sample size studies or novelty experiments, sometimes you get a hall pass, but still we all know that when we are about to publish a study, maybe the significance is all that matters, maybe from the reviewer perspective or in the current publication system. So I do think there has been some discussions about switching to Bayes factors or different statistics approach, but for these exploratory studies, I find it very difficult to figure out where to draw the line. So if there are any insights about this, I would like to hear more about it. Yeah.
Well, I think about the sample size and these exploratory studies that we had so far, there are a few things related to that I believe in the future it will change. First, talking about FNUs, which is the technique that I most work with, it's kind of new. If you look, when I started my PhD a few years ago, and I looked, I Googled FNUs, there was almost nothing there. And I was like, where am I going? Why should I do this? Because there was almost nothing. And thank you to Dr. João Sato. He was like, FNUs is a good thing. Let's gonna try it. And then now, almost 10 years later, we see a lot of people doing FNUs, and how many things that we were not able to do before that we can do now, such as look into the brain classrooms, look into the brain of, for example, children with Down syndrome or children with other kinds of conditions that we can do now. So in terms of the sample size, I believe that we have the combination of having new techniques with the pandemic, and the pressure of the publication. So it's a set of troubles that we have. So maybe it's not about changing the statistical way of looking to the data, but probably in the future we're gonna have more, we can improve these problems in future research with those techniques. So I think it's like a system problem with the all pressure to publish, to get report for the funding, so we don't have time enough to collect data from more patients or for more participants, and then so on. So it's new methods, okay, yeah, it's good, but old methods is to work, I think.
I think one thing to sort of build off of the exploratory part is that, we publish exploratory research within a system, and part of that replicability part of that is, it's sort of actually reliant on other people reproducing it. So if you had one person and you're like, this one person, and there are studies with one person, but it's usually like case reports or something like that, or where you can have, they're unique in enough of a way. So if you run something with 5 people and you find some result, and then maybe someone else can run it with 10 people and find the same result, and then you have a, sort of like a meta Bayesian like statistical thing where through the combination of this you do have 2 studies that say that blank. And then of course effect size is a big part of that, but it's not just the published effect size, it's whatever the real underlying effect size is. So if you were to go and a lot of studies that have been presented here have had some, not every study, but the language ones have all involved at least the auditory cortex in some manner. And if you published anything with FNIRS and the auditory cortex, people would expect to see activity in the auditory cortex or something like that, and they would not be surprised if that publication contained that information, which speaks to the effect size of that. And you can say similar things for the motor cortex and other stuff, and some of the other associative areas where either the brain activity is nebulous or the concept that you're looking at is nebulous. Like if you're like, what is curiosity? And you're like, I don't know what it is. And everyone will have their own definition for it. But I think that you're right that legacy like statistical methods can set that way. And you're also right that like newer methods could also be like kind of exciting and do that sort of thing, but they exist within that replicability system. The problem is that people don't want to publish the same thing that somebody else published because somebody else did it. And so now you're de-incentivizing the ability to improve upon that, effect size for whatever it is. And so you kind of lose a little bit of that because you're muddying the water. Maybe someone else says, this area is the part of the brain that's responsible for calculus, for instance. And someone else goes, well, we did it, but with algebra. And now they're not exactly the same study anymore, but if you were willing to be like, they're both math, then maybe the system can still work. But I think replicability is key there, and new methods may help, and old methods also work, as long as you're using them correctly.
I think I have some suggestion for that because we recently published a paper, and the main result we use the SPM and FDR correction in the Waskow level have a significance. But for our exploratory analysis, we do have the P lower than 0.5, but it is not past the multiple comparison issue. So I suggest that in that case, looking on the personalized features, and you can see what's the difference. Maybe specific participants have the low value or higher value, so we can try to interpolate why that's happened. In our case, we are investigating the autism, so they have the higher heterogeneity that happens. So I suggest to go in detail to the person level to check why that thing happened. That would be very interesting for the society and for the audience and the readers.
Okay, so I just wanted to bring it back to the weird populations. I really liked what you said about that. And I'm excited about the fact that, the Oura rings and Fitbits and actigraphy trackers are so commonplace now that I think that could be a really good opportunity to appeal to a wider variety of people. So neuroscience in general as a topic is daunting. And if we can destigmatize it and use education as a tool as we diversify neuroscience and have social media to help explain to people what it really is, I think we could really have a much better sample. Even working with veterans, a lot of our veterans are not comfortable with the healthcare system and are not big fans of technology, it's scary. And just being able to explain that, oh, it's actually just like your watch, except it can do these new things. I think as we keep having wearables and having them look more simple and more accessible, that this is a really, really great opportunity for the field.
Like just related to the weird population point in the mobile brain body imaging conference, the topic of what different sensors can measure in different demographic groups is actually something that worth paying attention to. So like EEG sensors that are developed to adjust for different hair types, or say like certain population groups being excluded because of some physiological features, like eye trackers with Asian eyes, not a good fit. So these things actually are like pretty awkward to talk about, but I think it's kind of worth thinking about it and be mindful of like what kind of population you're excluding from your designs. Yeah.
Yeah. Well, we have a great panel, so thank you, all of you, for your comments, and thank you for organizing this. I think.
We don't have much to say. Every time we organize, this is the second time we're doing this, but when we think about this panel, we know we need to do it because we want to hear from you, and then we're amazed at how time fills up and you have so much to say, and it's so resonant with what everybody else is saying. So thank you very much for capturing, I think were all the little conversations that were happening at the conference, which each of you got a chance to summarize. We're really, really happy that you're here, and I think you've all said it really well. You bring different perspectives. You're talking about reproducibility. You're talking about sharing your protocols. You're talking about making the studies more diverse and more real, and not just a narrow population. I mean, these are things that we hope the next generation of scientists will pick up on and take forward, and you talked about all of those things today. That was just amazing. What do we have to say, David and Matthias? You're here. This has been a really amazing conference. I think the second year we thought, what should we do to make the conference interesting? And it wasn't just about neuroscience of the everyday world. We thought we should think broadly about neuroscience in the everyday world. And as you noticed, we had a lot more clinical talks and a lot more wearables. And it's amazing how, again, aligned so many different talks were in terms of what they were doing in terms of data collection, which you all did a great job summarizing. So I think that what we're also learning is the everyday world is about brain, it's about behavior, it's about applying to clinical populations, it's about understanding what makes somebody improve or not. It's about that early diagnosis. It's about taking ecological tasks that are more relevant, that are more naturalistic, and we seem to have captured all of that. I don't know if you want to add to that, both of you. You should. No, you're good. David? We're really, really humbled to hear all the talks at this meeting and your great summary of what was discussed here. I think the only caution I would say, or at least maybe not put a negative note on this, is the idea that you said, small samples and not publishing these stunning studies. I think that it's okay for us to be in this newer field of doing ecologically valid studies. They're hard to do, they're messy, and everything does not have to be perfect. You don't have to have the perfect statistic yet. I think what's important is that balance between what you're trying to study and how applicable you want it to be in the real world. We're all here because we want our work to be applicable in the real world, and It's a little bit about that implementation in the real world that comes with its baggage, and we're talking about the baggage here, and that makes us all develop better methods. So in 5 years, I don't think we'll be talking about the same problems today, as you all would have found solutions to that, but we'll be talking about other things in that process.
And I just want to say, I heard you guys say, and I heard many other speakers say throughout the last 2 days that it's been really wonderful just getting this group of people together who are from many different fields who usually don't meet each other at conferences, and I think we've all learned a lot from each other. And for me, that was really a big goal of the meeting this year and last year. So thank you everybody for contributing to that success. And I think now we can officially close the conference. So we've all deserved rest after an intense 2 days. So
thanks to the IT team. Really, thank you guys. It was amazing, flawless, and I don't know, people were tuning in and out, people were walking in and out, people came in and then became virtual, and you guys just handled it really smoothly. So thank you, IT, thank you, facilities. This was, and thank you for the weather. You have to thank Boston for the fantastic weather that you got here. So thanks, everybody.